I'm Sue. Thank you so much for joining us today. I'm Director of Researcher and Community Engagement at Digital Science, and I have the utter privilege tonight of being joined by Venki Ramakrishnan. Venki is a Nobel Laureate. Venki is what we like to call the scientific triple threat. You were a physicist by training, you're a structural biologist.
Not for long.
Well, long enough. Long enough to get the badge. And you won the Nobel Prize in Chemistry. So you're sort of the Zac Efron of science.
Which is really molecular biology.
Yeah.
So I'm neither a chemist nor a physicist, but...
I love it. It's me claiming not to be a physicist. I do that quite a lot. You're a former president of the Royal Society as well, but now you're a group leader for the Structural Studies Division at the MRC's Laboratory of Molecular Biology. This is a tiny fraction, though, of what you've done and what you've achieved. So I wonder, can I start with the hardest question of tonight? Can you tell us a little bit about your background and what brought you to writing this book?
Oh, well, okay, so I spent most of my life studying the ribosome, which is the large molecular complex that translates our genetic information into proteins. And it's all the thousands of different types of proteins in each of our cells are made by translating genetic information, and that's done by the ribosome. And that's really what I was best known for. And it turns out, though, that protein synthesis has a lot of things about it. It's highly regulated. It controls the quality of proteins. And all of these processes break down when we age. So although I don't work on aging myself, the process I study turns out to be of central importance to aging. But the reason I wrote this book is that I was sort of aging adjacent, in the sense I was in a field closely related to aging, which had implications for aging, but not part of the aging community myself. And it's a big existential question. You know, humans have worried for centuries about, you know, why is it that we grow old and die? Why do some species live for days or weeks and others live for hundreds of years? So this is a long-term existential question. And for most of our existence as a species, and by the way, I don't think animals are aware of mortality. They're aware of death. They know when one of theirs has died. They even mourn their dead, some of them. But they don't know that there's an expiration date like we do. And so this is a big question. And for most of our existence, there's nothing we could do about it. You know, the saying goes, inevitable like death and taxes. But in the last 40 or 50 years, biology has made great strides in understanding many of the underlying causes of aging. What actually happens to our molecules and our cells and organs as we age, and what leads to death. And along with this, there's been a huge explosion in aging research. So I should say for quite a long time, it was sort of a backwater in the molecular biology community. And, you know, people, mainstream molecular biologists didn't really work on it. And it's only in the last 40 or 50 years that that's changed. And part of it is because we're all facing the issue of an aging society where people are living longer. And so there's a great imperative to make sure we're healthy as we age, and we're healthy in our old age. And so there's a lot of money going into it. And at the same time, there's a lot of private investment. There's 700 companies, startup companies doing some aspect related to longevity. Some of them are selling all sorts of supplements. They vary from very, very serious, based on rigorous science, all the way to the other extreme. And then at the same time, there's an influx of a lot of private money going into these companies. A lot of it from California billionaires, who I suspect, I mean, if you ask them, they say they're interested in helping humanity and they want to improve human health, etc. But I suspect that, a lot of them are middle-aged men worried about growing old. And that's generated a huge amount of hype in the field. So you get, extreme hype, people who think, we're going to live for hundreds of years, and the person who's going to be 1,000 years old has already been born. That's another statement. So I wanted to take an objective look at the field. And because I don't belong to the community, I don't have any financial interest, nor do I indeed have a vested academic interest. None of my grants depend on, aging research. So I can afford to be, quite frank about calling it the way I see it. So, if I offend, people in the aging community, it's okay. They're not part of my community. So, it's my effort to take a frank look at the field. And also, humans are curious, so the hope is that it'll satisfy people's long-standing curiosity about this, eternal phenomenon of aging and death, this real existential question. And at the same time, it'll give them the tools to interpret new information as it comes along. Next time you read a news item which says, new cure for aging, or, this, that, or the other, you can place that in the context of the fundamental biology that I hope that the book will, expose them to.
It is an incredibly emotive topic, and you can kind of understand why. I even struggled to say the D word just now when we were chatting just outside the lecture theatre, because it is quite anxiety-inducing and yet entirely inevitable.
Yeah, humans have always, had this complex about death and the finality of it. I think we've evolved to want to live, partly because if you don't want to live, it's not very efficient. You won't pass on your genes unless you struggle and try to live. And even our language, we don't say somebody died. We say they passed on, or they passed away, or they departed, as if it's some transition, to some other station in life. And if you look at most religions, death is viewed as a transition. It's not viewed as an end. It's, either you go to heaven, sometimes you corporeally arise, like in the Abrahamic religions, or you have your body may decay, but your soul is immortal and occupies some other body. But the idea is that you somehow live on after you given up this particular body. So I think a lot of human culture is about denying the inevitability, or the finality anyway, of death.
Yeah, you talk about the philosopher Stephen Cave and his 4 coping strategies, but really your book is this romp through the latest advancements in molecular biology and the iceberg of research on which they sit upon to really address that first one, which is how do we extend that life as long as possible? Yeah,
Stephen Cave had a very interesting idea. He said, we adopt 4 strategies to deal with death. And plan A is simply not to die. And plan B is that you die, but you can be corporeally, resurrected. And that's, a lot of the Abrahamic religions used to traditionally insist on burial, and they didn't like cremation because of that. And then plan C is, well, you can, your body may decay, but you have an immortal soul, which can occupy other bodies or have an existence of its own. And plan D is you live on through your legacy. And the legacy could be your works or monuments you build. You endow buildings after yourself or build pyramids. Or it could be through your offspring. So you can have biological legacy as well as, other kinds of legacy. But, Woody Allen said he didn't want to live on in the hearts and minds of, people through his works. He wanted to live on in his apartment. So the inevitable. So I think in the past, people would treat all these plans as as equally plausible. But after the Enlightenment and sort of more the growth of rationality, I think people are more and more converging on plan A, which is simply to try not to die. And that's what, aging research is really about, staying healthy. I mean, the more sane end of the spectrum is not about extending life, but it's about staying healthy for a longer fraction of the life. And of course, then the other extreme is to simply postpone death indefinitely.
I think that's really interesting because it's one thing to be alive and it's another to be able to live. But really these definitions of where life begins and ends can have huge consequences politically and ethically and morally. And you touch on that in your book at the start.
Yeah, it's a little bit, we think of life and death as very definite singular events, but neither of them is extremely well defined. Birth, of course, highly controversial when a human starts to exist, and we can see that in the abortion controversy in the US, and even if you think of something that starts at conception, even the moment of conception is not very well defined. It's not right when the sperm meets the egg. All sorts of things have to happen before you get a fertilized egg that, starts ball rolling, but the fertilized egg has to implant, and it has to grow, and only at some point does it develop some sort of sentience. So it's all complicated at that end. At the other end, death used to be defined in lots of ways. People used to define it as just, okay, if you didn't have a pulse, you were dead. And then, people looked at electrocardiograms. But then they found out that you could restore people who had apparently no heart function by CPR. And then, many states started moving towards brain death as death. But of course, people have gone into coma sometimes for months, sometimes even for years, and eventually some of them have come out of it and some of them die. And in the US, there's which is very federal, there are peculiarities. There was a young woman who died in California according to the laws of California, but her relatives in New Jersey didn't accept that she was dead because she's not dead according to the laws of New Jersey. So they forced California to ship her body to New Jersey, and then she was in New Jersey for a while, and then eventually died according to the laws of New Jersey. And you could have a weird situation where in one case somebody died and it's perfectly valid to donate their organs to transplant recipients. In another state, that same thing would be committing murder, if you did that. So you're right, neither is well defined. And the other interesting thing is that when we die, most of our cells are still alive. Entire organs are still alive. That's why you can donate them to transplant recipients. And so what do we mean when we die? I think what it means is we can no longer function as a coherent individual. It's that ability to function as a coherent individual that's lost when we die.
Okay, I think everyone's anxiety about death is pretty high right now. So I think we should maybe talk about some of the amazing research that has been going on, and some of it quite interesting research in the very British definition of the word, that you cover in your book. But we do like a definition here because this is the Royal Institution. What I would love is if you can just help us along with some of the terms that you use in your book, because I think in society we sort of use a lot of these interchangeably. So we have lifespan, life expectancy, and crucially health span as well. Can you expand on those terms a little bit more?
Yeah. So life expectancy typically refers to life expectancy at birth, which is when somebody is born, what is the more likelihood, what age are they likely to live to? And that life expectancy has gone up from below 40 about 150 years ago to over 80 in most developed countries. And then it's actually plateaued off in the last decade or so. That may be due to other problems such as obesity or other effects. But anyway, that's life expectancy. And of course, life expectancy at birth is not the same as life expectancy at a different age. So if at birth, you might be expected to live at 80, but if you've already lived to 80, then you can be expected to live another 10 years or something, you know, so it's not quite fixed in that sense. Lifespan should more accurately be called maximum lifespan, and that's asking how long, if everything goes well, you don't get run over, you don't get eaten, you don't die of disease, and everything goes well, how long can you possibly live? And this is what I meant. Some insects live, a mayfly lives only a day, and you know, a giant tortoise lives almost 200 years, and there are sharks and whales that live even longer. So that would be what I'd call maximum lifespan. And then health span is really about what are the number of years of healthy life that we have. So if life expectancy is 80, what's the sort of health span? You know, what fraction of those 80 years are we going to live healthily?
Mm-hmm. So really, we're aiming for that healthy life, you know, the living part of being alive. Now, with your work in ribosome research and really understanding how it makes proteins, how it reads that genetic code that is within all of us, you have played this fundamental part in, let's face it, practically every biological process that goes on in the body. So you're very well placed, really, to talk about these things and evaluate some of the challenges we're facing.
Yes and no. I mean, I'm probably better placed than a non-science or science journalist who has an undergraduate degree or something like that, because I have spent a lifetime doing molecular biology in a field that has some, you know, as I say, has some central implications for aging. But as I said, I'm not an aging specialist, and there are many areas of this book which are highly specialized. For example, stem cell research, or you know, or even things like epigenetics. Although actually I did work on epigenetics once. But you see what I'm saying? Science is highly specialized, and so it's a fact that almost every chapter in the book could have been an entire book or many books in itself, even at the popular level. And in fact, there are popular books on epigenetics, and I've cited some of them. There are popular books on you know, the gene and you know, basic DNA molecular biology. So I think it's highly complex, and what I've done is to do my best as a kind of informed scientist to take a look at the literature and you know, try and distill it. And I've also taken a lot of advice, you know, so you know, one of my colleagues at the Royal Society, who was the vice president for biological research, is Linda Partridge, and she's a very famous aging researcher. And so I would constantly pester her with emails about this or that and ask her what she thought of it. Now, I should say, I don't want people to think that these are her views, because I wouldn't want to saddle my views on her, because there are times when I saw things differently. But she was a very useful kind of guide to sort of help me navigate, you know, these 300,000 papers that, you know, were published in the last 10 years or so.
So for a lowly chemist like me, and maybe for people that are not as familiar or haven't done any biology, maybe since they've left school, also like me, can you give us a little reminder of what we're talking about here? You make a really beautiful comparison between a big city like London and a cell, a living cell in a living organism. Can you tell us a little bit about that analogy, what that means, and the complexities and challenges that brings?
Yeah, so death is really the accumulation of damage. Oh, well, sorry, aging is the accumulation of damage over time. Largely, I mean, it's mainly chemical damage that affects the molecules of life, but then, filters upward to more complex systems. For example, to the collection of molecules in the cell, to organelles, components of the cell, to cells themselves, and then to collections of cells that form our tissues and so on. And at some point, it manifests itself in these symptoms that we all recognize as, when a guy like me looks in the mirror, I can recognize, the symptoms of time and age. And so that's what, aging is. But at the molecular level, it has many different aspects, all the way from, our genes and DNA, all the way to, more complex systems. And so to get back to your point, I compare it to a city. If you walk around a city like London, you take the amazing choreography for granted. For example, there's an electric power grid that supplies, electricity here. There are sanitation people who remove all the waste, from London. There's water supply that provides you clean water. There are restaurants that provide food. There are power stations that generate all the power that you need to run a city. And then there are transport systems. There are arteries, there's, the London Underground and buses which carry goods and people from one part of the city to another. And there's something that has to keep all this coordinated, something we can think of as a government, but it's not just government. It's institutions have to keep, and the institutions have to talk to each other, and everything is coordinated. And it's not obvious, it's sort of almost invisible, it just happens. You only notice it when things start breaking down. And it's when that coordination starts breaking down, then you get a problem. So, for example, if the Royal Society or Royal Institution suffered a power cut, most people in London wouldn't notice, okay? But if the London Underground stopped working, then a lot more people would notice. And if, let's say, the government of London suddenly stopped working, then, you would suddenly have chaos, and you would not be able to function. And I think the cell has many similar elements to it. There are lots of things in the cell. There are powerhouses that generate energy, which are our mitochondria. There are this sort of command and control, which is our genetic program, which resides in our DNA, which you can loosely think of as a very democratic government. And then, there are things that traffic molecules from inside the cell, different parts of the cell. The cell has border control. It allows certain things to come in and not others. And then it also has an export system, which, it exports stuff. It has a garbage disposal system. So when you have accumulation of defective molecules or dead molecules, the cell has to get rid of them. So there are lots of things that are very similar. And these are the things that break down as we age.
So as we scale up from the cell, we have all of these complex processes going on just in these single cells and lots of potential points of failure or degradation. As we start to scale up to organism size creatures, size really matters, doesn't it? You use a beautiful quote from another Nobel Laureate, Rabindranath Tagore, about the butterfly counts not months but moments.
Oh yeah,
time enough.
Actually I might have a slide. Here we go. So, there's the mayfly, an insect, lives for only a day. The monarch butterfly is unusual because it can have a life cycle where it lives for months and another life cycle where it only lives for weeks. They have the Galapagos tortoise. There are tortoises which have been, tracked at about 170 years. It's very hard to really have accurate records, but that 170 years means a tortoise could have been around when Darwin was actually observing it, so that's an interesting thing. And then you have a whale that lives over 200, and and a Greenland shark that lives 400. And then what you just talked about is this rough correlation between size and lifespan. So all the smallest animals are clustered at the bottom left, and the largest animals are at the top right. That's a general rule, but of course you can see there are exceptions. Now, why would that be? Well, it turns out this is a deep evolutionary principle. It doesn't mean that death is programmed and that each species has a program that says, okay, if you're a mouse, you get to live for 4 years, and then this program kicks in and it's going to kill you. Rather, what has happened is that evolution only selects for the ability to pass on your genes. And if you're a mouse, there isn't much point in spending a huge amount of resources into maintaining and repairing the body so that it ages, doesn't age and lives for a longer time, because it's much more likely it's going to be eaten or starved to death or die in a flood or something long before it ever can age. On the other hand, and so in that case, it's better to spend the resources on rapid growth and reproduction, and that's what mice do. On the other hand, if you are a large animal, then it pays to live longer because it gives you more time to find a mate or mates and more time over a longer period to have more offspring, because longer animals also have a longer gestation period. So in each species, there's been this fine balance. Now, if you look at the top left, you notice a bat. Okay, now bats are about the same. That Brandt's bat is tiny. It's about just as big as a mouse if you didn't look at its wings. So why do bats live so much longer? Well, that again is the evolutionary principle. Bats can fly. So not only can they escape predators more easily, but they can forage widely for food. So their chances of starving are also less. And so, it's true that as a rule, flying animals like birds or bats live longer than animals of the same size that are land-bound.
I remember reading this chapter and thinking, as a woman of 4'9", this is not boding well for me. But actually, within species, it pays off to be smaller, doesn't it?
So for example, they've done this extensively with dogs. So where dogs have a huge variation in size, from a Great Dane to a Chihuahua or something. And so it turns out small dogs live longer than large dogs. And this has to do with the growth hormone pathway, the same thing that allows large dogs to grow fast and to a large size, causes them problems later in life.
Mm-hmm. Heard it here first. So I was quite pleased to read that, I must say, as a tiny woman. I do want to talk, just before we move off from this graph, about human life expectancy, because that has doubled in the past couple of 100 years.
Yeah.
And what is that down to?
Yeah, so if you look at this chart, and I can actually, so there's human, and that's the dot that represents a human, and they're well above where they should be on the line. That means we're doing very well for our size. And that's because, well, it's for two reasons. One is that intrinsically we are doing, as a species, we are doing better, but we've also dramatically expanded our lifespan and life expectancy. And that's the increase in life expectancy, which is the, probability that you'll survive to some age, the likely age at which you'll die. That's improved because of many reasons. Most of that increase from about 35 to 40 years, about 150 years ago, to over 80 years now, so more than a doubling. That's happened mainly because of advances in early life, and that is, we have dramatically reduced infant mortality and also early childhood mortality. And this is a result of better public health. I mean, London is one of the pioneers in that, because the finding by John Snow of the fact that cholera was transmitted by water from a contaminated pump led to London being one of the first cities to separate sewage from water supply. And that alone improved, the odds dramatically. And then came vaccines, and then later on came antibiotics. So over the years, we've really improved our odds of surviving to the point where we can reach adulthood. Now, even in Victorian times, if you managed to reach adulthood, your chances of living to 60 were not bad. So it was really that you had a great chance of not surviving up to that point. Now, we've also made advances at the other end, and that's because we've better learned to tackle diseases like diabetes, which used to be essentially a death sentence if you got diabetes. And then we also were able to treat many kinds of cancer. So, for example, my mother-in-law died of lymphoma, not many years after she was diagnosed. And my wife, on the other hand, was treated in 2010 and is still in remission. So, cancer treatment has dramatically improved. Heart disease, we've dramatically improved our treatment of heart disease, even prevention like statins. And so I think we've also made improvements at the other end, and that's, improved life.
You talked about cancer. It's really another difficult topic to think about, to talk about. But it features quite heavily in the book because a lot of the processes that lead to aging of cells and of organisms are in this fine balance of enough repair and regeneration, but not to the point where it gets out of control. Can you tell us a little bit more about that?
Yeah, so when the cell is stressed, the cell sends it into one of two directions. Either it commits suicide in a phenomenon called apoptosis. And apoptosis, by the way, occurs naturally during development. It's an essential part of life. But it can also occur as a response to various kinds of stress, including DNA damage. And the other is to send the cell into senescence. And I'll come to senescence in a minute. But the point is that these processes evolved so that you wouldn't get an aberrant cell that would become cancerous. We have trillions of cells. If we kill off a few million that are problematic, we would never notice. Cells in us are dying all the time. We're just completely unaware of it. And so we wouldn't notice that. On the other hand, if that cell survives and becomes cancerous, then it's a real risk to the entire organism. And so evolution has really selected for pathways that take care of this problem by sending the cells into either senescence or to suicide. The senescent cell is a cell that doesn't divide. It's not functional in the way a normal cell would be, for example, if it's a cell of the immune system or if it's a skin cell. It's not doing its normal thing. It's sitting there and it's secreting inflammatory compounds. And the reason it's doing that is to attract the immune system to the site, because it's typically the site of an infection or a wound, and it's signaling to the body to come and repair the damage and clean it up. And in the process, the senescent cells are gotten rid of. Now, as we age, what happens is those processes themselves are less efficient, and we start accumulating senescent cells, and then we get a problem of general inflammation because these cells are secreting inflammatory compounds. But you have to remember the origin of it may have been as an anti-cancer mechanism early on in life. And this sort of interplay occurs throughout the book. I mean, well, throughout, not the book, but the aging process, that many things that, prevent cancer early in life actually are causes of aging in one way or another. And this, again, is that evolutionary principle, because evolution cares about whether you make it long enough to pass on your genes. It doesn't care too much what happens to you afterwards.
Mm-hmm. You talk a lot about cell regeneration, cell division you've just talked about. Cells generally have a finite number of times that they can divide, and the discovery of why that is, I think it was Elizabeth Blackburn, discovered, you're going to have to forgive my pronunciation, is it?
Telomerase.
Telomerase.
Yeah.
Wasn't sure. Just a chemist here, just a chemist. And what's really interesting about that is it is offering a way where we might be able to not just better understand it, but start to get involved and maybe start to control some of the cell division.
There are a number of scientists who are very gung-ho about telomerase. Let me back up and explain a little bit about the issue. So when cells divide, our DNA molecules have to divide. They have to essentially double, and then each cell gets a copy of the DNA. Now, our DNA is a long molecule, so it has two ends, and then it's a linear molecule. And because of the way that DNA divides, the ends of the DNA are not replicated each time. And so each time the cell divides, the DNA gets a little shorter, okay? And that's in most cells, in normal, differentiated cells. But of course, if you have special cells which are not only dividing themselves, but they're responsible for regenerating tissue. In fact, when we develop for the first time, you could think of the fertilized egg as the ultimate stem cell because it results in everything in the human body. And so that's called totipotent stem cell because it can give rise to the entire body, but also to the placenta. But then as you later get to the embryonic stage, each of those cells, if you isolated them, they could develop into any kind of cell. Later on, the stem cells get more specialized. They can only, let me just... Yeah, so this is just showing you how a fertilized egg can develop into a human being. But if you now go to watch that process at the cellular level, at the early stage, these cells, each of these cells could actually develop into any kind of cell. But later on, they become specialized stem cells. So for example, some stem cells can only give rise to cells of the blood system. So the immune system and red blood cells. Others can only give rise to skin or hair. Others can only give rise to bone, cartilage, and connective tissue, etc. And others give rise to cells of the nervous system. So you can see how these stem cells are differentiated. Now, what happens as we age is that we start depleting these stem cells. Many of them become senescent, they become damaged. The remaining stem cells are the clones of a very few stem cells, whereas before we had a diversity of them. So all of these are problems. So the question is, could we take these final cells and persuade them to go backwards a little bit? And then they could then help regenerate tissue, because otherwise we're having trouble, regeneration of tissues and so on. And I would have thought this was somewhat unlikely, but somebody named Yamanaka, whom you mentioned, figured out how you could take these cells and make them go all the way back to the early embryonic state and make essentially what are called pluripotent cells, which can differentiate into any kind of cell. Of course, when they take these cells and start growing them in culture, you also get tumor growths, because some of these factors are actually oncogenes. And so how to control that, so you only have, proper development like the natural embryo and not have these, cancer-causing side effects. That's a tricky problem. The other is that, you want to take them back only a little bit and not take them all the way back. And that involves giving the Yamanaka factors transiently and then turning them off somehow. And that's what people are trying to do. This whole field of cellular reprogramming is very, very exciting. There's a lot of work on it. And it's not just about aging, because people also want, as you get old, your cartilage wears out, people get knee problems, shoulder problems, etc. And if you can regenerate cartilage, you'd be better off. You could perhaps, for diabetics, regenerate some of the key cells in the pancreas. Of another sort of long-term thing is if you have damaged heart muscle, you might be able to regenerate muscle. And ultimately, could you regenerate neurons, after a stroke or something? That, that's very long in the future. But regenerative medicine is a big, huge industry designed to tackle many of these problems. I mean, if we get an arm cut off, we can't regenerate it, but a starfish can. So that's the sort of difference. And so it's a big problem, and not just related to aging. And it's a complex problem of how to do it safely and effectively and in a targeted way. So although there's a lot of excitement, I as an outsider, I think it's an enormously challenging problem, and I personally don't think that it's going to happen tomorrow. I mean, there's a lot of hype about it, but I don't think it's going to happen tomorrow. I think they'll make progress, and we'll see where they get with animals and figure out how to do things.
I'm going to talk a little bit about proteins again, given your interest in this field and how much of protein research features in this book, in this summary of the aging process. I wanted to pick up on the quote that you used from an Ernest Hemingway novel, How Did Somebody Become Bankrupt? Gradually and then suddenly. Because a lot of aging can be this accumulation of things over time. One such thing, we talked about cells being like cities and organisms being like cities with all these processes to maintain things, bring resources, but also eliminate waste products or things that are not working as they should.
Yeah.
And proteins play rather a large part in this. In fact, the biggest killer of people, the cause of death in Wales and England is dementia, and that is caused by a quirk in protein folding and the accumulation of that.
Yeah.
Can you talk a little bit about that?
Sure. So, going back to the city analogy, imagine a city where nobody got rid of waste and garbage. Such a city couldn't really function. It's certainly not a city as densely populated as London. And so the body has the same problem. We make proteins, but proteins, even in the normal course of things, are made when they're required, but they're also destroyed when they're no longer needed, because often it's not advantageous. It's actually deleterious to the cell to have a protein hanging around when it's not required. So some proteins are required all the time. They're what are called housekeeping proteins. They're required for the basic functions of the cell. But others are required at particular points, for example, in development or during a response to some environmental cue, etc. And you don't want them around. The other problem is proteins acquire defects. Partly proteins themselves get old, they get oxidized, they stop working the way they should. They start unfolding. So proteins are long chains. And in fact, in this lecture theater, David Phillips once gave a Christmas lecture showing how a lysozyme molecule, it was in a model, it was about this big, and then if he unraveled it, it went all the way to the ceiling of this exact theater. And there's a very famous picture of this, of the lysozyme polymer unraveled. And so if a protein unravels, then it has a tendency to stick to other protein molecules and form large tangles. And one example of tangles are these tangles or filaments found in the brain cells of dementia patients like Alzheimer's. Their neurons are full of these tangles of, there are two kinds. There's a tau filament, which is inside the cell, and then there are these beta amyloid filaments, which are all over, especially outside the cell. And these are all the result of our own proteins misbehaving, and they're partly the result of the inability of our body to get rid of them, in a timely way. And in fact, one of the machineries that gets rid of these defective proteins is called the proteasome. It basically feeds in the protein into itself and chops it up into its little components, which are called amino acids. And if you have defects in the proteasome, that actually does make you more likely to develop some of these diseases of aging. So, and then there's a large organelle in our cell called the lysosome, which the city analogy would be the big garbage recycling center, the city dump where things are sent and then they're recycled there. And it's more efficient, though. It doesn't just accumulate junk. It actually does recycle them. And so things are transported to the lysosome if they're defective. In fact, entire organelles that are defective can be moved to the lysosome for destruction. And if that process is, not functional, then you can also get aging. So these are all things to do with recycling and a lot of it to do with proteins.
And actually the body deals with these things in a really fascinating way, using these biomarkers, things like ubiquitin, for example, to almost, it's the equivalent of spray painting a cross on a diseased tree almost, and those get recycled.
Yeah, no, it's a good point. So with proteins, or with unfolded proteins, one of the classic ways that the body deals with it is it senses that this protein is defective and puts a tag on it. And the proteasome then recognizes the tag and then targets it for destruction, or other things take it along, for eventual destruction.
So by better understanding some of these mechanisms, maybe around biomarkers, for example, could we expand that research to almost start to create early diagnostics for these potential diseases that can impact our healthy lifespan? The need for markers for things like dementia and unfolded proteins is pretty high. And I think, I talked to a colleague of mine, Michelle Godert, who's an expert on tau and also its implications, and really many neurodegenerative diseases. And he pointed out that one of the most useful things would be to have early biomarkers, and they are being slowly developed. There are also other biomarkers for aging. For example, as we age, we also accumulate tags on our DNA. And, there is some debate about whether these tags are actually the result of aging or the cause of aging. And of course, the more strong advocates believe that it is actually a causative thing, that the tags themselves change the genetic program in a way that causes aging, such that if you were to remove the tags, you would reverse some of the effects of aging. And that whole field is a big field in its own right, and that's part of the cellular reprogramming. Much of what that does is to reverse some of these tags. And that's not entirely what it does, because it actually works with transcription factors which affect
the program of which genes are expressed, but they also, some aspect of it has to do with these reversal of these tags. And but the tags themselves can be thought of as a marker of aging. I mean, one thing we hadn't discussed is how do you, if you're developing anti-aging treatments, how do you even know whether your treatment's working or not? Because what are you going to do? Wait until the person, 20 years until the person develops symptoms, and then those are very subjective and they can be highly variable, and so on. So there's a need for developing markers, and people are developing markers. And one marker based on these tags is called the Horvath clock, which is named after the guy who lives in California who discovered it. But there are other markers which also use the same tags, which are similar. And then there are other markers in our blood which are indicative of aging. And so the hope is that the community will arrive at a consistent set of markers that everybody can agree that this is a real marker of aging. Because time is not a good marker. We all know 70-year-olds who are very spry and can run around, climb mountains, or do whatever they want, and others who are in not very good shape. Okay, and so we age biologically at different rates, and what we really want is to ask what is their biological age, but there might not be a uniform biological age for each person, because your organs may have all aged somewhat differently. There's no such thing as, oh, you're this single number. So I think the phenomenon of measuring aging is becoming more rigorous, and hopefully will improve our ability to assess not only aging but also the effect of therapies.
Mm-hmm. I think the rigor and the integrity of assessment and evaluation is so important, and is really that defining factor between science and some of the pseudoscience that you talk about in your book that is prevalent in this area where people really are desperate to try anything.
Yeah.
You talk about the contradictions in some of the research. Is red wine good for us maybe this week, maybe not next week? Sorry, Dad. Depends what week we're in. You talk about antioxidants, how the role of the mitochondria may be involved in sort of trying to reduce the number of free radicals that we have that could be potentially damaging, but then other studies show that, what doesn't kill you makes you stronger. Where are we going with this? Where is the sort of...
Yeah, I think, this is true of any field of science, that at the very frontiers, things are always a bit fuzzy, and things you thought were really happening turn out not to be true, and other things are true, or things are true but not for the reasons you thought. So I wouldn't say the aging field is particularly different, except because it gets hyped so much. People often have painted themselves into a corner, and then they don't want to give up their own ideas and so on. And maybe that's just human nature, because Max Planck once said that new ideas in science don't emerge because people with the wrong ideas change their minds in the light of new evidence. Rather, they just die, and eventually some new generation looks at the evidence with fresh eyes and moves on. And it's been paraphrased as science marches one funeral at a time. And so aging research is really no different. You mentioned free radicals and oxidative damage. I think there's no question that oxidative damage and damage from these reactive oxygen species. See, our mitochondria are where oxygen is used to oxidize sugars. So it's effectively where we burn sugars to make energy. You can think of it that way, okay? Because you're producing CO2 and water, just like you would if you took sugar and lit a flame to it and burned it, okay? So, except we're doing it in a very controlled way at 37 degrees Celsius, and and extracting and using that energy in a useful way, not just as heat. We're actually generating useful energy out of it, useful chemical energy. But that's where oxidation occurs, and if that oxidation is not complete, we get these partially, reduced oxygen molecules, which are called reactive oxygen species, and they're highly reactive, and they can damage our DNA, they can damage other parts of the mitochondria, they can leak out and damage other parts of the cell. And so there's this big thing, and people have showed, that this is actually a cause of mitochondrial damage and of aging. And so, I think if you go to any health food store today, you'll find loads of antioxidant supplements, okay? And that's, so you can see there's some solid science behind it, but the fact is that most of those supplements were shown not to work. And why is that? It's because maybe those supplements are not being used by our body in the where they're needed and in the way that they're needed. Okay, so it doesn't mean that reactive oxygen species are not a problem. The other thing is, of course, when we generate a small amount of reactive oxygen species, our body senses that. They're also signaling molecules. They're not just damaging molecules. We use it, our body uses it for signaling various chemical, sending chemical signals. And so our body also induces repair mechanisms. And so they have the paradoxical effect of actually improving things in smaller doses. But of course, in larger doses, they could be still damaging. So I think this just shows you that you can't, you've got to take the field in all its complexity and really understand what's going on, and not just say, oh, these things cause a problem, so we should go take antioxidants. That's not a great solution.
Venki Ramakrishnan, thank you so much.